Hello, Mr. Allman here, and this is your second installment of the Allmancast tour of the cell. As you learned about in the cell theory, all cells are the basic unit of life, and so each cell needs to be capable of carrying out all processes necessary in order to maintain life. One of the most important functions within the cell is the way in which matter and energy are processed. Matter needs to be brought in, needs to be broken down, converted in some way into a usable form so that energy can carry out all of the different movements and building and growing and reproducing that a cell needs to do. So our main topic for today is going to be the mitochondria. We'll touch a little bit on the chloroplast, however, because that's only found in plant cells, the details for chloroplast is going to be left to our unit on botany. So first of all, let's consider energy. Energy is defined as the ability to do work. It's hard for us to be able to come up with a very rigorous understanding without really using your imagination, because energy is something that can be measured, it can be observed, but more often it is observed as it's transferred from one place to another. One thing that you should remember from chemistry is that energy is going to be stored in chemical bonds. So there is going to be a net release of energy when large molecules are broken down and those bonds are reformed into smaller molecules. Now that ability to do work is going to allow the cell to carry out a lot of important things. So finding food and taking that in, breaking molecules down, building new molecules like proteins and lipids, making ATP an important energy currency within the cell, removing waste, reproducing, and regulating its internal conditions. All of those things certainly require the use of energy in order to carry them out. Now, energy is going to be processed within the cell, just like matter is processed within the cell, but neither of those things are made. No cells, even including plant cells, make energy or make matter. Rather, they change and transfer and rearrange those things into forms that's going to be more convenient for the cell to be able to use. Now, for most cells, they're getting their energy from food or from the sun. If it's an autotrophic organism, it's using energy from the sun to be able to build organic molecules like sugars, whereas heterotrophic cells are going to take in food and use those molecules as a source of energy, and we'll get into that in a moment. There are some exceptions to that, such as the chemoautotrophs. Those are the bacteria that are able to digest some of the hot organic molecules that are spewing out from lava vents, and that's what forms the base of the food chains in the deep ocean. But for the most part, these two things can summarize most forms of life on Earth. Now, the two organelles that are primarily responsible for processing this energy are going to be the mitochondria and the chloroplast. Now, the mitochondria is going to be found in nearly all animal cells, including nearly all human cells, whereas the chloroplast is going to be an organelle that is responsible for carrying out the steps of photosynthesis, and this will be found only in plant cells and plant-like cells. Now, there's a reciprocal relationship between these two. Carbon dioxide and water are necessary ingredients in order for photosynthesis to occur, in which that matter is going to be converted into sugar using energy from the sun. Now, that sugar or other organic molecules, when combined with oxygen in the mitochondria, is going to produce a lot of heat as well as a form of chemical energy called ATP. So the process here is called cellular respiration, and specifically when oxygen is available, it's called aerobic respiration. And the details of this complex biochemical reaction, again, is going to be covered in our metabolism unit, whereas the details of photosynthesis will be covered in our botany unit. One term or acronym that you've heard before, at least in this video if nothing else, is ATP. Now, ATP stands for adenosine triphosphate. Adenosine refers to two parts. It's a nucleic acid connected to a sugar. The triphosphate is the part here in blue and gray, and this stands for 3 phosphate groups. Those phosphate groups consist of phosphorus atoms and oxygen atoms. So this molecule of ATP has bonds that can be quickly and easily broken within the cell, and so it provides a much more convenient source of quick energy for a lot of the basic processes that need to happen within the cell, moving things, breaking things down, activating, and shutting things off. So ATP is going to be the main product of what the mitochondria are producing. So let's look now a little bit more at some of the specific parts that are responsible for carrying out these processes. First, in terms of matter processing, are the lysosome. The lysosome is going to look like a fairly simple structure. It's going to be really a membrane-bound sac that contains enzymes. When you look at the electron micrograph, which is the picture taken through an electron microscope, you see the lysosome as this darkened, shaded circle. You can't see a lot of detail inside because those enzymes are things that are too small to see even with this type of microscope. What's going to happen is that the lysosome that has those enzymes inside is often going to be merged together with a structure that contains food. And so this is a way to be able to utilize those enzymes and have them act upon and break down that food and then take that digested food onto the mitochondria, which can then use those broken down molecules as a way to be able to produce ATP. So this is a kind of internal cellular digestion, a way for the cell to be able to break down molecules in preparation to be able to convert them into alternate forms of energy. So they digest large molecules. They're also helpful in terms of being able to digest damaged parts because those enzymes can also be used to be able to break down anything within the cell. So if there's another organelle that is worn out or damaged in some way, the actual physical material that part was made up of can be broken down, and those atoms can then be recycled and reused to make new working cell parts. In the most extreme example, these enzymes are used to actually digest the cell itself because there is a limit to the lifespan of a cell. And after a certain number of divisions, the cell needs to go through a process called apoptosis. And apoptosis is going to be a type of programmed cell death where all of the parts of the cell are going to be broken down, and then that matter will be reabsorbed by neighboring cells, which will then take its place. Those enzymes that are within them work best at pH 5 for reasons we're going to investigate in our biochemistry unit. Here you can see a picture showing a cell undergoing apoptosis. The one on the right here, each of those little spheres is a little part of the cell that is in the process of being broken down, and those eventually are going to bud off from each other and those parts reabsorbed by neighboring cells. There are diseases that actually impact the lysosomes. These are often fatal because those digestive enzymes, if they don't work, then the cell is not able to effectively break down wastes. It can't recycle some of those materials, and cells aren't going to die when they are supposed to. Tay-Sachs disease is an inherited genetic disease that affects the lysosomes in the cells of those who are inflicted by it. And so in this case, there are undigested fats that build up, and those undigested fats are going to become waste that is going to take up space in brain cells, causing the death of those cells, mental retardation, and death at an early age. The peroxisome is another type of membrane-bound sac that you should be aware of. This is going to be responsible for things like detoxification, breaking down fatty acids, and it's going to contain a variety of enzymes and chemicals such as peroxide, which the cell is going to utilize in the process of breaking those things down. It's important that in all cases with lysosomes and peroxisomes that all of those chemicals are kept within these membrane-bound sacs in order to protect the rest of the cell from those digestive enzymes or the peroxide, because you don't want those things harming parts of the cell that are helpful and necessary for the cell to keep. Like the lysosomes and peroxisomes, vacuoles are also membrane-bound structures. In many cases, a vacuole is much bigger. It's still a membrane-bound sac, but its purpose is mainly going to be storage, storing things like food, water, enzymes, ions, things that the cell might need in the future but doesn't need yet, and are too important for the cell to just get rid of. A vacuole can be a place where it can be stored temporarily. The mitochondria is going to be one of the most important organelles that's found within cells. Its structure, it's made up of two membranes. There is an outer membrane that gives it a kind of a kidney bean or a pill-like shape, and then a highly folded inner membrane. All of the folds of the membrane are going to help to increase the surface area of that inner membrane, because it is within the mitochondria that a series of important chemical reactions called cell respiration occur. The cristae are the name given to the folds of that inner membrane. This is going to divide and create two different spaces. There's going to be the matrix of the mitochondria, which is that center space that's inside the outer membrane and inside the inner membrane of the mitochondria. There's a second space that is in between the two membranes, and that is called the intermembrane space. There's also a small amount of DNA present in the mitochondria, and that has to do with the origin of mitochondria in terms of our theory of where it is that these organelles first came from. You can see on the right, this structure is clearly a mitochondria because you can see the folded inner membrane. This electron micrograph is going to be a slice. It's a slice of the center part of the mitochondria, and you can see the majority of the space is made up of the matrix, and then you have the little tiny bit of space that's present there in between the folds of that inner membrane. Now, the function of the mitochondria is going to be aerobic cellular respiration. So respiration is a catabolic process where food molecules are broken down, creating a large amount of ATP, carbon dioxide, and water. Carbon dioxide is a waste, and so that is going to diffuse out of the cell and eventually leave either the cell or the body of a multicellular animal. That water may be recycled for other purposes, but the ATP is the main product. ATP is the thing that the mitochondria are really built to do. They're producing this molecule, which is then sent throughout the cell as a sort of energy currency that can be used to accomplish all the other functions necessary. Almost all eukaryotic cells have mitochondria because they all need energy. And so in order for that cell to keep going, it needs to rely upon those mitochondria to produce more ATP. So you should expect that more active cells are going to have more mitochondria. So think about, for example, a heart cell, a cardiac cell, cardiac muscle cell is going to have a huge number of mitochondria because that is a cell that is working constantly 24 hours a day for its entire life, allowing you to beat 1 to 2 times per second. So the number of mitochondria is going to be much greater in this type of cell or other types of cells that are constantly working. Finally, I'll touch briefly on chloroplasts. Chloroplasts are going to be a structure that's found within plant cells. When you look at a image here of the chloroplast, you can actually see the greenness that we see in plants comes not from the cells themselves, but from the chloroplasts that are within them. Because a leaf is going to be chock full of cells that are just stuffed with chloroplasts, because this is where photosynthesis is happening. Chloroplasts are going to be similar in size to a mitochondria. In both cases, these are organelles that are about the size of a bacteria. So they are certainly, they're going to be less than 10 micrometers across, something in the area between 0 to 5 micrometers on average. Thanks as always for listening, and please let me know if you have any questions.